Our today's topic is macroevolution and the tree of life, and in which we will be discussing about the cladistics. We have discussed a little bit about the cladistics in the previous lecture, but we will be more discussing about the cladistics and how do we apply this approach of systematics to study the tree of life. Now, first of all, we will discuss that what were the early scientists who first thought about it. Willi Henning was an eminent German entomologist, and he stressed the need to develop a new and more objective method in the systematics, which has come to be called cladistics. So this was the first scientist who knew about the different groups of organisms, and he knew that what are the characteristics which are defining them to be in a particular clade, and what are the different characteristics which are not giving any information. So uninformative characteristics and informative characteristics, he knew about those, and he gave rise to the science which is called cladistics. So in the cladistics, the fundamental aim of cladistics is to identify the clades and to discover, reconstruct the tree of life, right? So we are working to construct the tree of life with the help of the branches that we are acquiring through the different organism. Every species is a branch, and we study all the species together that will construct a tree, and that tree is called the tree of life, and that we can do through the cladistics. And in the cladistics, the patterns of relationships are shown in branching diagrams or cladograms. And the example of cladogram is this, right? So here you can see the two parts of the diagram. One is A, another one is B. So A and B, one of them is a correct representation of the cladogram. So this is reconstructing the phylogeny of vertebrates by the cladistics methods. In the A are the defining features of vertebrates, the position of bones, skull, and tail. Then of course not, right? So the vertebrates do have other characteristics, and what are those? In the B, the cladogram is much more defined on the basis of tail in the chordata, and vertebrates are bone and skull. Not all the vertebrates have the skull, right? And not all the vertebrates have bone. For example, the shark does not have bone. These are cartilaginous fishes. And salmon and frog, they do contain the skull, but the frogs, again, amphibians, do not have the tail. So you see that how the cladograms are refining the thing or the characteristics for us. So now if you see that where the two branches are emerging, that is called node. The most closely related species are joined most closely. If two nodes are together, that means they are joined closely. And the branches in the cladogram joined at branching points are nodes. So where these two branches are emerging, those are called nodes or branching points. And this marks the base of a clade. So where from the clade starts, that would be the base, and that is called node. So now apomorphies. There are some characteristics which were, Henning has his own way of giving names to the characteristics. So Henning called the phylogenetically informative characteristics the apomorphies or derived characteristics. Apomorphies are the features that arose only once in evolution. So what would be an example of apomorphies? Apomorphies is a characteristic that is present only in one species, or the characteristics that is the characteristic of only one clade, right? So for example, Psittaciformes is the family of the parrots. So they have a particular characteristics of a curved beak. So that is apomorphic characteristics. So this is how we can, identify a clade based on a single trait or single characteristic. So apomorphies shared by two or more species are termed as cynomorphies. The cynomorphies of order Psittaciformes include the deep hooked beak or the unusual feet. So that beak is not only characteristic of a single species, but also more than one species. So when it is happening, we call that as synapomorphism. Now, there are some older distinction as well. You may already be familiar with those, just like homology and analogy. So, homology, when there are two characteristics that are same in two different species. And analogy is when there are the same characteristics is performed by the two different organisms, by the different organs, but in the same way. So, vertebrate limbs, wing of a bird and paddle of a whale, arm of the human, all are homologous. They are similar in structures. But the paddle of whales and seal, although they are doing the same thing, they are helping in swimming, but they are analog. They are not homologs. So, and after that, the cladistics, what makes it more interesting that now we have the molecular evolution. There was birth of molecular biology in 1950s and 1960s, and we were able to know more about the organisms at molecular level. So now that molecules include the RNA, DNA, and proteins. And these molecules are similar if the organisms are similar, and these are different or differently evolved if organisms are far apart in the tree of life. So comparisons of molecules allow analysts to do two things: to draw up the trees of relationship and to estimate the time. So not only we can draw that how much these two organisms are related, but also we can see that how much far in the past they are sharing a common ancestor. So the trees of relationship which are made on the basis of molecular analysis, we call them dendrograms. These can be based on a comparison of amount of difference between the protein sequence, and a best fitting diagram, dendrogram or branching diagram is drawn. So we see the two proteins from the two related organism and see that what is the difference between the both. Just take the amino acid sequence and align that amino acid sequence with the protein of the other species and see that how much they are having in common. If they are more common than a third organism, then they are more related. And also you can infer that if there are more changes, they are far having the far common ancestor, and if they are more related, then they are having the common ancestor in the near past. And the time estimation comes from the concept of molecular clock. The amount of difference in the fine structure of protein is proportional to the time since they last shared a common ancestor, that we have already discussed. And the nucleic acid sequence must be aligned, right? For example, you know that A, G, T, C are the four bases which are making the nucleic acids. And if you align the DNA and RNA sequences together, then you can see that who two organisms are sharing the same common DNA or RNA, right? So normally we do that with the DNA sequences. We are aligning them and see that which one has, more related, right? So if I am having the DNA of a banana and DNA of chimpanzee and DNA of human, if I align those DNAs, then through alignment means that I put the DNA of one organism over the other and see that where they have some commonalities. So I would see that banana has less common DNA with a human than chimpanzee. Chimpanzee is much more common in DNA, and that is done through the computational techniques, and we can, see the similarities and differences in silico as well as in the laboratory experiments. And in this way, we can study the macroevolution and tree of life. And once we have developed the whole tree of life, we would be better able to understand the life.